Welcome back to Acing AP Biology. In this video, we will finish up cellular respiration, discussing its final stage, oxidative phosphorylation. Oxidative phosphorylation takes place in the inner mitochondrial membrane. It has two components, the electron transport chain and chemiosmosis. The carrier proteins that make up the electron transport chain are embedded in the inner mitochondrial membrane. Since each carrier is more electronegative than the next, electrons are passed down and energy is released little by little after each step. The final electron acceptor is oxygen because it is very electronegative. Most of the ATP produced by cellular respiration comes from this stage. The first electron carrier complex is flavin mononucleotide. At this protein, the 6 NADH from Krebs cycle and 2 NADH from the pyruvate oxidation drop off their electrons. As these electrons flow to the next protein complex with the help of ubiquinone, which is a small hydrophobic electron carrier, energy is released. At protein carrier complex 2, the 2 NADH from glycolysis and FADH2 from Krebs cycle drop off their electrons. They have to drop off their electrons at the second protein complex because they are of lower energy. The NADH from glycolysis comes from the cytosol, and so they give off energy when they have to enter the mitochondria. The FADH2 has less energy than NADH, so it has to come to the second protein complex. The NAD plus and the FAD molecules are then recycled back to glycolysis, pyruvate oxidation, or the Krebs cycle. Electrons continue moving down the chain. The third complex is composed of a chain of cytochromes and another electron carrier that helps transport the electrons down the chain is known as cytochrome C. At the fourth protein complex, oxygen is ready, waiting to pick up the electrons. It picks up all the electrons along with 2 protons to form water. Now, all the energy that was released due to the electrons moving down the electron transport chain is used to pump protons into the inner membrane space against their concentration gradient. This creates a proton motive force. Protons then move back down their concentration gradient using ATP synthase, an enzyme. This process is called chemiosmosis because protons are moving back down their concentration gradient. The flow of protons through ATP synthase powers the phosphorylation of ADP to ATP. The net products from oxidative phosphorylation are 32 ATP, NAD+, FAD, and water. Now that we have gone over all the stages of cellular respiration, let's count up all the ATP that was produced. Glycolysis produces 2 ATP through substrate level phosphorylation and 4 ATP through oxidative phosphorylation. Each NADH molecule produces 2 ATP. So since there were 2 NADHs coming from glycolysis, 4 ATP are produced. Therefore, glycolysis produces a total of 6 ATP. Pyruvate oxidation produces 6 ATP as well through oxidative phosphorylation. The 2 NADH from pyruvate oxidation are of higher energy than the ones in glycolysis, so for each NADH molecule, 3 ATP are produced. Therefore, a total of 6 ATP molecules are produced in pyruvate oxidation. Krebs cycle produces 2 ATP from substrate level phosphorylation, 18 ATP from the 6 NADHs, and 4 ATPs from the FADH2, because each FADH2 produces 2 ATP molecules. So overall, Krebs cycle produces 24 ATP molecules. Therefore, cellular respiration in eukaryotic cells altogether produces 36 ATP molecules. 32 come from oxidative phosphorylation and 4 come from substrate level phosphorylation. That's it for this video. In the final lesson of Unit 7, we will discuss other ways ATP can be produced, such as fermentation. See you then. For more bio content, hit subscribe.